All right, so last time we talked about the history behind evolutionary thought. How did we get to Darwin's theory of natural selection? We talked a little bit about artificial selection, remember, which is where humans select specific traits, um, and then those traits end up in the offspring of those individuals that they select. We talked about Lamarckian evolution and why that is not actually correct through August Weismann's experiments that he did. And then finally we got to Charles Darwin. I started telling you a little bit about the background of how he came up with his theory. So remember, his theory, which is simplified to survival of the fittest, can be expanded to this description, which is something I kind of want you to memorize and have known by heart, because I think it's really important to have this background when you understand how natural selection works. So remember, individuals vary. That means they're different. That variation is heritable, which means that the things that make them different between individuals in a population can be passed on to their offspring. There's a struggle for existence, which, remember, Darwin found from Malthus, who said that there is a limited number of resources, and because of that, if the population gets too large, there's going to be a struggle for existence. We're going to die because of war, famine, competition, disease, all that stuff. And then because of that struggle, the individuals that have the best adaptations, characteristics that will enhance their ability to survive and reproduce, will mate more often and therefore pass the characteristics that are better on to their offspring. So Darwin thought, what if nature did the selecting instead of us humans, which we clearly saw could cause significant change over time with dog breeds and different breeds of cows and apples and things like that. Okay, so this process results in the change in frequency of alleles over time and thus evolution. So you want to make sure you know this by heart. Now, one of the common misconceptions that people have about natural selection is that individuals can evolve. That's not true. An individual cannot evolve. Only a population can evolve. A lot of people that don't understand evolution fully actually think that individuals can evolve, and that is not the case. So a population can change, but an individual cannot. So when Darwin got home from all of his travels aboard the HMS Beagle, which is the ship that he took to the Galapagos, he took years and years to come up with the background, the evidence to support his theory of evolution by natural selection. So he did years of compiling, years of looking at specimens, specifically as it came with his finches, which were a really well thought out, which we'll talk about later, a well thought out, well put together pieces of evidence to support his theory. He finally published his book, and he only published his book when he got a manuscript from Alfred Russell Wallace, who was another naturalist at the time. And Darwin was kind of a big naturalist by the time he published Origin, and he was well thought of. So this young naturalist sent him his theory, and it happened to be exactly the same as Darwin. So Darwin kind of freaked out because he thought that he was going to get beaten to the punch of coming up with this idea. Now, Alfred Russell Wallace didn't have all of the background that Darwin did. And all of the evidence that Darwin did. So Darwin finally decided that he needed to publish his book. The reason he waited so long was because the church was worried, or he was worried about the church's reaction and the public's reaction, even the scientific community's reaction to the release of such a controversial topic. That species were no longer created specially by God, but that they had common ancestors, and eventually through small incremental changes led to all of the different species we saw today. Now today, the origin of the species, or On the Origin of Species as it's called, but sometimes it's called Origin of Species, is probably the most influential book aside from some of the major religious texts that we have today, including the Bible, the Quran, the Torah, just because of its impact that it has had on everything after that point and how we think about medicine and the biological sciences in general. So now we have to talk about, like, how do we know that this process occurs? We said that, okay, evolution, the process makes sense, right? But do we have evidence that it actually works and it's feasible in the world? The first thing is we have direct evidence of it. we have evidence that things can evolve over time. One of the main ones that they love to focus on the AP exam is the creation of bacterial-resistant drugs, or bacterial resistance to drugs, I should say. So it's kind of outlined below that you can see a bunch of bacteria with one resistant individual. So they have a gene that gives them resistance to a bacteria. Now, when you add the antibiotic, that selects against the individuals that don't have resistance and for the one that does. So you can see here, because you're now creating an environment when before they were all competing for resources, you give this one bacteria all of these resources, it's going to reproduce like crazy. And what you see is over time, we end up creating a population that is completely resistant to that specific drug. It's also true if you look at guppies. There's because of intense predation of guppies, it can result in drab-colored males. Now females normally like males that are brightly colored. So they'll find a brightly colored male and want to mate with him more often. But what happened over time in this case was that males became more and more drab due to predation because they were more easily seen. So we can actually see the shift. We've seen this happen over the course of time. Once predators were added to a particular pool that had guppies in it versus one that didn't have predators in it, you'd see males that were much more bright in color. And those are just two examples of evolution that we've seen. There's many more. There's also the fossil record. So the fossil record looks at fossils over time. And the way we look at geological time is, as we'll talk about this in a second too, is that the oldest fossils are at the bottom, the rock layers that are lower, the more recent rock layers are at the top. And I talked about this before, that based on estimations, 99.9% of all species that have existed in the 4.6 billion years that the Earth has been around are currently extinct. And only 0.1%, which we estimate between 1 and 2 million species, currently exist. So that's kind of an amazing thing to think about, of all the different types of organisms that have existed in the past. Just so you know, if we ever talk about paleontologists, a paleontologist is someone who studies fossils. Ross Geller was a paleontologist. So how do we know how old the fossils are? Well, we do something called radiometric dating. Now, there is carbon dating, which usually is used to date younger fossils and younger tissues that are less than 30,000 years old or so. But we use other forms of radiometric dating to test older ones. So what it does is it uses isotopes of carbon and other elements to figure out and date rocks and fossils. Now I'm going to explain how it kind of works, but it basically deals with the half-life of these isotopes, and the half-lives can range from 0.7 years to 48.6 billion years, which is an insane, range. But these subtle differences in the properties and the subtle differences in the proportions of these two isotopes can actually tell us the dates of rocks. So remember, the half-life, if you remember back to chemistry, is the amount of time it takes for half of a sample to radioactively decay. And different isotopes decay at different rates, and that rate is very regular. So, for example, if it takes 10,000 years for one half-life to occur and 3/4 of the isotope is left, then it's dated to be 5,000 years old, because if half was left, it would be 10,000 years old. If there was a quarter left, that means you did a half of a half, that would be 20,000 years old. So that's the logic of how we kind of date rocks. This kind of shows you this, the logic behind how it happens too. I don't know why neutrons is spelled that way, it's really weird, but anyway, this is the logic of how it happens. I don't think they're going to ask you to do this on the test, but you never know. I think it's highly unlikely. What we usually use to date older rocks is potassium-argon dating. My daughter is currently going through things she shouldn't be going through, so give me 1 second. I'll be right back. Brinley, what are you doing, Pete? Okay, I'm back. So one of the other types of dating that we do is potassium-argon dating, and this is the one we use to date older rocks because it has a half-life of about 1.25 billion years. So, which kind of matches up with how long life has been around, which is around 3 billion years or so, 3.6 billion. So, since argon is a gas, all argon will leave a sample of rock while it's magma. And once it solidifies, the gas will produce, the gas will kind of be trapped, and then potassium-40 will decay. Then, because the gas is trapped, the potassium-40 decay product will actually stay there. Depending on the concentration of potassium-40 and argon, you'll actually end up figuring out what the half-life of that is. Now, this isn't, you can't use this for sedimentary rocks, right? But we can use it to figure out the age of certain layers of sedimentary rocks if they're found in the same layer, both magma and that layer. So that's how we do it. The law of superposition is basically the idea, as I mentioned before, is that older rocks are at the bottom, younger rocks are at the top. Now they can flip due to some geological processes, but we know ways of figuring that out. Specifically, geologists know how to do that kind of stuff. So then once we know the date of the layer, we can figure out the date of the fossil, because the fossil was obviously formed during that layer. When sedimentary rocks form, basically when something dies near a water's edge, it gets kind of buried and then gets buried by the water and then buried by some sediment. And eventually it's not broken down by bacteria or anything. It has to kind of be in an anoxic environment. And then over time, it'll slowly turn that organism into stone, which is what a fossil is. One of the interesting things that we notice is that complexity definitely builds significantly over time as we look through the fossil record. For example, nothing more complex is ever near the bottom, but there are still less complicated things obviously at the top, but you can see complexity building through the fossil record, with the newest layers having the most complex organisms in them. Another really strong piece of evidence, and you want to know this term, is homology. And this is where the term homologous chromosomes comes from. Homologous, it doesn't mean that they actually have the same genes on them. That's not what homologous means. It means that they're evolutionarily related. So homologous structures are evolutionarily related structures, and a lot of times those structures don't necessarily have the same function in the other organisms. So if we look at all of these mammals at the bottom, you can see the different bones in the arms of a human, a horse, a cat, a bat, a bird, and a whale. Now, in a human, obviously, those bones are used to pick up and manipulate the environment. That's what our arms are used for. In a horse, it's used for running. In a cat, it can be used for walking and running as locomotion, but also can be used as a weapon. Bats use those same bones as a wing. Birds use them as a wing. A whale uses them as a flipper to guide them through the water. But they still have the same underlying anatomy. Just there's been slight changes in that anatomy that has resulted in different functions. So homologous structures are evolutionarily related, but they have different functions in organisms. And that's one piece of evidence that we have of evolution. There's also embryonic homology, which is fascinating. So if you look at the embryos of all of these different vertebrates, which means that they have a backbone, these embryos don't look that much different between a fish, salamander, turtle, chicken, rabbit, and human. At that stage, they're all very similar. You probably, unless you were an expert, couldn't tell the difference between them. But slowly, after development, we have significant changes. And those significant changes after development have a lot to do with these specialized genes called Hox genes, H-O-X. And we're going to talk about in a separate video how development kind of works. I go through, the logic behind how genes get turned on and off, starting from one cell all the way through a complete organism. That's going to be a separate video. But you can see here that early on, really there's not much difference between any of the vertebrates, even though when they are all adults, there's significant differences between vertebrates. One of my favorite types of evidence is actually vestigial organs. These are organs that really don't have a function or have much of a function in us or in a species that currently exists, an extant species. Extant means alive, extinct means dead. But in the ancestors, they had a very important purpose. So our appendix is an example of this. Now, it doesn't mean that an appendix is not important. The appendix in us, they think, though we don't need it, is used to repopulate our gut with good bacteria after we have bouts of dysentery or diarrhea. So it's important for that function, but it's not really necessary. We don't need it. In our mammalian ancestors who ate only plant material, it actually was an important place where the bacteria that helped them to break down cellulose lived. So we cannot break down cellulose. Remember, it has beta-glycosidic bonds between the glucose molecules, but some bacteria can, and there's organisms, or there's mammals that are only plants that have a lot of those types of bacteria in their guts, and it tend to be harbored in the area of the appendix, which was much larger back then in those, and including in species that are currently vegetarians or herbivores. Whales actually have floating hip bones that do absolutely nothing. They have like a false little pelvis there. They have a tiny little femur, but it has shrunk so much, and it really doesn't have that much of a function compared to the other structures, compared to us specifically. Now they have their flippers, but their legs are completely gone. So they have those floating bones just kind of sitting in there. Here's showing you what I was telling you about herbivore organisms. Like, look at their appendix. The koalas is huge. All it does is eat eucalyptus all day. Right? The zebra has like a storage tank. Rabbits have storage tanks. The humans is much tinier, as you can see here. All right. Now, once we discovered in the 1950s that DNA was the genetic material and the structure of DNA, at that point, that could have thrown a complete wrench into our idea of how evolutionary theory works, right? So it was a nerve-wracking time, I guess, for evolutionists when we discovered DNA. Because what you'd think is that species that have these tiny differences, who have less differences, will probably be more related. Ones that have bigger differences would be less related. And if we would have not found that to be the case, that would have thrown a complete wrench in our idea of how evolution worked. Okay, so what we ended up finding is that is absolutely the case, that it fits perfectly, that species that look similar and seem related tend to have similar molecular homology. They have similarities in their DNA sequences. All life forms use DNA and RNA as their instructions for making proteins, which suggests that DNA was present in us and a common ancestor of all living things. And what we can do is, if we compare DNA and amino acid sequences of closely related organisms, they'll show higher levels of similarity than those that are distantly related. And we can actually do some sequence comparisons, which we're going to do in a lab right at the end of the year. And we can compare the differences between humans and chimps, rhesus monkeys, cows, mice, and rats, as you can see here. And you can see they're all very similar, pretty much, with the human and the chimp being very similar. But sometimes you have tiny little changes that make them slightly different. Dashes mean they don't exist in that organism, so it's moved over one. But still, they're very similar. And we're all mammals, rats to humans. So we should have very similar DNA in a lot of places, not so similar in others. Another cool piece of evidence for evolution is called convergent evolution. So natural selection states that the environment does the selecting. So sometimes unrelated organisms that are not, like birds and bats, for example, right? They both have wings, they both fly. But the development of those characteristics were independent of each other. They don't share evolutionary usage, I should say, in the past. So in convergent evolution, unrelated organisms will develop similar characteristics due to selective pressure. Selective pressure is like how the environment affects them, or similar environmental challenges. So, for example, the penguin, which is a bird, it no longer flies or uses its flippers to fly. It lives in the Arctic and some in the Antarctic. The same thing's true with the dolphin. They look very similar. They're streamlined shaped, their ability to go through the water quickly. The dolphin was a mammal that developed this. The penguin is a bird that developed this completely independently, not evolutionarily related structures. Their flippers of the bird are not evolutionarily related, at least in terms of their function, to the flippers in a dolphin, but they do have a similar shape because it allowed them to move through the water better and allows them to survive better in those in particular environments. That should say streamline, not steamline. I need to change that. If anyone's ever seen the old Batman, that's funny. If not, it's probably not funny. Or it's not funny at all, I don't know. These structures that develop through convergent evolution are called analogous structures, as opposed to homologous structures. Remember, in homologous structures, they're structures that look different, but they share evolutionary history. In analogous structures, these are structures that look the same, have similar function, but they're not evolutionarily related due to the environment kind of selecting for and pruning those particular structures. Okay. Biogeography is also really cool. So if you look at marsupials, which are one of the classes of mammals that exist, marsupials and this is wild. If you look at all these things, they look like different mammals that would be found on the mainland. So the gray-headed flying fox clearly looks like a bat. Possums are possums, but koalas are like little bears. The wallabies are like small, like there's kangaroos and wallabies. There's the kyoko, which looks a little bit like a little rat or something like that, or some sort of rodent. The sugar glider, which looks like a squirrel that flies. The dunnart looks like a mouse. Okay. The wombat looks a little like a capybara, if you've ever seen those. The quoll looks like a cat almost. But what's amazing about this is all of these individual species are more closely related to each other than they are to anything that looks like them on the mainland, like on the other continents. So like the gray-headed flying fox is more closely related to a koala than it is to a bat, even though they look the same. So this is showing you examples kind of convergent evolution, but there's other factors as well, which we'll talk about, like adaptive radiation, which we'll get into later. So I think that's super fascinating. We also have plate tectonics, which were found kind of in the early mid-century, that the plates actually, in the past, the continents have moved and shifted dramatically, and they still are shifting, and they're moving due to convection currents underneath the Earth's surface in the mantle. So the idea of continental drift, which is not technically true, plate tectonics is true because they're sitting on plates. It's not just the continents that are moving. At one time, around 200 million years ago, the world was a supercontinent where all of the continents were kind of in contact with each other. Well, how do we have evidence? How do we know that? Well, the first thing is South America looks like it jams right into Africa's armpit. It almost fits perfectly in there if you look at the coastlines of the two. But on top of that, we also see that there's fossil evidence where South America has the exact same fossils in it as Africa, even though it's thousands and thousands of kilometers away, exactly in the area where it fits into the armpit of Africa, which is so fascinating to me. So this is just another example of some of the evidence that we have through the fossil record of why and how old the Earth is, but also mostly through how old the Earth is, I should say. Another cool thing is endemic species. Endemic species are species that are only found in one place on the Earth. The Galapagos has a lot of endemic species, particularly because it's so geographically isolated from everything else, because it's an island way off the coast of any of the mainland. So endemic species form as a result of geographic isolation, and they're unique. Like, for example, at the Galapagos, there are these aquatic, or they're swimming, why can't I think of the name of them? What's wrong with me? Swimming iguana. They're these giant tortoises that are huge. Lemurs exist only not in the Galapagos, but lemurs exist on Madagascar, which is in a similar situation. So when you get these islands that are far away from the mainland, you tend to get these species that only exist there and nowhere else on the planet. And one of the really interesting things that I learned when I was in college, I took a course on island biogeography and the evolution on islands, is that organisms that are tiny and go to islands, like from the mainland, so they're tiny on the mainland, tend to get larger on the islands due to a lack of predation. So being larger actually helps them to survive better. But things that are very large on the mainland that end up on islands end up shrinking, like pygmy elephants and pygmy rhinoceroses and stuff like that. So it's a really interesting thing, because for those individuals, being too large actually limits their ability to survive on an island because there's not as many resources. So being smaller was of benefit to them. All right, so that's it on evidence of evolution. If you have any questions, let me know.